In both, let's be prepared. I'm here. I am Chaeyoung. I am not in.
No, I know you are you. I'm just.
It's going to connect. I can. Mm. Uh,
All right.
yeah. Hey, that's me. Okay. So, okay, cool. So welcome to my very scientific presentation about my project. So it's called Gone with the Smoke, Decoding Airway Epithelial Responses to Climate-Related Inhalants. And my name is Chaeyoung Kim. So, wood smoke. We all know that wood smoke is bad, but we tend to overestimate the power of wood smoke. So in 2023 alone, Canada burned roughly 15 million hectares. So that's nearly 7 times the historical average. Smoke from those fires exposed an estimated 354 million people and across North America and Europe. And a recent Nature study estimated about 5,400 acute deaths happened, linking into the resulting PM2.5 and 64,300 chronic deaths in North America and Europe just because of the fire that happened in Canada. And in Ontario, just 1 week of wildfire smoke in June 2023 was estimated to cover over $1.2 billion in health impacts. So apparently all these numbers, now you know that it's not a minor seasonal nuisance, but it's actually a really huge evolving problem, like public health problem. So I'm going to go in a little detail on why wildfire PM2.5 is very dangerous. Okay, so we all know what PM2.5 is. We've heard of it, right? But these particles are often very, very small, especially when they're first emitted. That means like for the same mass concentration with different like chemicals, there's actually a large number of particles and a lot of reactive surface area. So it's not just about like how much smoke is in like the air, it's about how biologically active they'll be when it enters into your like body, in your lung, because it's so small and there's so many. And more importantly, when we think about like forest fires, we only think about, okay, so it's like wood burning and it's going to me and I'm like drinking it. But it's actually more complex because as wood smoke travels, it's going to travel through so many pollutants like air allergens and like even UV light from sunlight. And how far that it travels is very important in human health. So when we say PM 2.5 wood smoke, it's actually more very diverse and complex than that. And also there's a recent study that as it moves, they tend to stick together to form larger particles. So it's even more dangerous than when it first occurs. Anyway, so this I brought because I thought this was interesting. So this is a very recent article. And this is actually a case of an 87-year-old man developing severe respiratory distress after inhaling forest fire smoke. So basically they did bronchoscopy because he was having like respiratory problems. And then they found out there were like these carbon bronchial casts that was covering his lung, which were making his, have very respiratory symptoms. So when the cast was removed, his breathing improved. So this is a dramatic example of direct airway injury and obstruction caused by wildfire smoke. So now we're informed about the wildfire smoke, but the literature is very limited right now because until now people only like scientists only investigated like one thing, maybe like just wood smoke or just like diesel exhaust smoke or just like cigarette smoke, right? But we never actually combined them and figured out what's going to happen to our body. So that was my research question. So wood smoke, it's not like we're just standing in front of a tree and as it's burning, it's going to travel long distances to you. So what's going to happen when smoke overlaps with other climate-related exposures, such as air allergens? So basically, so that is my project, my 2 years. Characterize the transcriptional response of the airway epithelium to climate change-related environmental exposures. So I have two hypotheses. So first one is exposure to wood smoke and aeroallergen or their combination produced distinct transcriptomic responses in area epithelium. And my second hypothesis is that wood smoke and aeroallergen co-exposure induce synergistic gene expression changes in human bronchial epithelial cells. So I mean, like you guys might like think that this is like obvious, aeroallergen wood smoke bad, but there aren't like much research that's done about this. So that's why I'm here. So this is like a roadmap of what I'm gonna do in my lab, but I'll go in a detail. So first, we will utilize human bronchial epithelial cells collected and banked during research bronchoscopies from research participants with respiratory conditions and healthy controls. So they're provided by the Carlston Lab. And then to incorporate EDI principles, we are going to assemble a cohort balanced for age and sex and diversity and ancestry. So yeah, we'll all be like noting them down. Anyway, so human bronchial epithelial cells are differentiated in a air-liquid interface. So you can see that there's only media on the bottom and there's like cells on the top on the apical side. And that's because we're trying to mimic what's happening in the airway. So that air passes through. And just so there's only media on the inside and cells on the outside. And that actually helps cells differentiate into a more bronchial-like state than just putting them in like a submerged culture. Also, when in this way, when we expose them, the aeroallergen would first interact onto the apical side, which we want to imitate. And then we bring them to a very specialized facility in like right here, like to be honest, it's like 500 meters from this building. So I first expose them to ragweed allergen, and then I bring them to a special facility to smoke them with wood smoke. And then cells are exposed directly for 2 hours using the in vitro exposure system with wood smoke with PM 2.5. And then we're trying to simulate what's going to happen in like a condition of a local forest fire, not just random fire. Yeah, and then they'll be harvested, and then total RNA will be extracted. We're also doing transepithelial electrical resistance, which is checking like the barrier resistance, and also, yeah, basically mapping out a library. So this is actual photos of my lab, like when we expose them. Yeah, so that's like a custom built furnace exposure system, which is the only one in Canada right now. And yeah. Also, my rotation. So I did my rotation in the Lim lab, thanks to Dr. Emilia Lim, and they were doing anthracosis. Yeah, sorry, anthracosis project, but they were also kind enough to show me how their RNA sequencing ways and help me do a literature review. So I actually had to go to their lab meetings and present this chart. So I made this chart, which is comparing total RNA sequence and poly-A tail RNA sequence. Lucky for you guys, I won't be explaining this all. So just see that I did them. Yeah. Yeah, so basically, because I have to do sequencing at the end of my, at the later stage of my project, and I have I only took like one course of computer programming, so I have no idea what dry labs are. So it was a very good opportunity for me to get exposed to dry lab component and also actually understand what I'm going to be doing in my project. So, and then this is a chart that I made for myself. So my experiment, basically integrating what I learned from my lab rotation. Yeah. So these are my references, and thank you.
Okay, very good. Issues, there's a group in here at 12:45. That's why I'm rushing a bit. Sorry. Okay, Jennifer. It's fine.
Thanks.